And I got a leave meeting message. I will ignore that.
Yeah, leave me, ignore that.
Okay. And now I lost my train of thought. That happens a lot these days. And as John mentioned earlier, I must be the oldest living member of the society. Well, I hope not, but that's better than being the oldest dead one, I guess. We looked at the seasonal changes in its biochemistry and so forth, and published a few articles on it. And I got an NSF grant to continue that work, and continued to study even after I got my PhD in Alaska for about 10 years in the winters and summers, travel back and forth between the university. Well, it got to a point where that was, really taking its toll academically. In other words, the universities weren't very happy with me disappearing. And right after that point, I had a 5-year program in the Antarctic. And so I would disappear for a few months at a time, as would my lab. And so we settled down to looking at, again, the same phenomenon, but in more temperate species of insects, and comparing northern and southern latitude species of happened to be a dipteran or a fly, goldenrod fly. And that was interesting in a lot of ways, but in addition to just glycerol that produced sorbitol, fructose, and as the Arctic beetle did too, and I didn't really appreciate at the time, fair amount of trehalose. And so overwintering insects also relied on trehalose. And if one began to look at the relationship of these various agents, there was a lot more to it than just quantitative indicators of how much was present at any given temperature. Because many of the freezing tolerant species would not survive particularly low temperatures. Like the Arctic insect would freeze at -10, was happy at -40, at -60 it would die. And of course, -60 was an extreme temperature, but nonetheless reachable in the interior of Alaska. And it was the same for the other species that I looked at. Well, I reached a point in the late 90s where I was becoming sort of frustrated because I could see a junction ahead in my career. There are more species in the world that I could look at for eternity. And some of my colleagues were doing that. And that wasn't challenging because it was the same story, but the numbers were a little bit different. And I one day got a phone call, and it was a recruiting agency looking for the vice president of research and development of a new company being formed called Cryomedical Sciences, which had two technologies. One was a quote unquote blood substitute solution that could be used in injured patients who were exsanguinated, lost a lot of blood, and had their body temperatures lowered. And the other was a cryosurgical technology. So I joined the company, which supposedly was for 3 years, and they paid the university, and everyone seemed happy to get rid of me. I was trying to manage both projects and needed help. And Mike Taylor gladly agreed to come from Cambridge to Pittsburgh, where we were managing the blood substitute program. And he led that and developed a solution known as hypothermosol. I took over the mechanical aspects of developing liquid nitrogen-based hardware, cryosurgical devices, with a graduate student of mine, Xiao Cheng. And we spent those 3 years and actually continued up until a year or two ago doing that kind of thing. Around late 90s, son John, was finishing his work at Cornell, and he took an internship in Florida at Mote Marine Labs, studying, you guessed it, manatees. Made no sense to me. But anyway, his girlfriend was a marine biologist, wannabe, which explained it. But he came home and one day, and from Florida, I think, if I got the timing right, and indicated he wanted to spend the semester reading. And I knew that was BS. So I said, no, you come to the lab and you work. And I got him in a lab with a colleague, Rob Van Buskirk, who was working on hypothermia. And he moved into that lab and began to study cryobiology, and took Mike's invention of hypothermosol and put it through various iterations and came up with CryoStore, which is now one of the principal standards for cryopreservation within the cell therapy arena. And I, on the other hand, continued devices, and something I never thought I'd be doing, but was assisting in all kinds of surgery across the US and Europe in cryosurgery. And that basically covers the territory. Did I leave anything out?
No, that's wonderful. Penny, our puppy heard your voice and she climbed on my lap and she's staring at the screen, wondering where Grandpa is. So there's a dog in my lap at the moment, and it's not a lap dog. So anyway, I figured since it was Grandpa talking, I couldn't say no.
You allow dogs in your office?
I'm actually at my house.
Okay.
I flew in from the West Coast and landed at 3 a.m. this morning, so I didn't get to the office yet. But,
okay.
Well, that's that. Thank you so much for that background and introduction. There's a couple little tidbits that Big John left out on some of his history. What he also failed to mention, that while he was an undergrad, he was also a college athlete and an avid swimmer, very good swimmer. So that's he was very busy in his undergraduate career as he pursued.
The highlight of that is in my first and second year, I was on the wrestling team, and the wrestling team finished second and third in the nation those 2 years. Had absolutely nothing to do with me. They went off and left me, but I can make that claim. I was in a championship level wrestler.
Oh, that's great.
I was cannon fodder.
There you go. Well, you know what they say in the movie Grease: if you can't be an athlete, be an athletic supporter. So while you were in Alaska, I think this is another interesting thing that if you could just spend a minute or two on, you did some work on the pipeline as they were putting the pipeline in. What was that like, and what did that involve?
Well, there's two memories. Oil had just been discovered in Prudhoe Bay, the north slope of Alaska. And I woke up that morning and saw it on the local news headlines. So I went to my friend, fellow graduate student Wayne, and I says, we got to move quick and get some stock in British Petroleum, which made the discovery. And he said, okay, well, none of us knew anything about stock. We went to a stockbroker, took all our savings, which wasn't that much, and bought stock. Little did we know the rest of the world knew that days before. And so we bought stock at its high point. And I kept it for years and actually sold it at a loss. But no, so to get from Fairbanks to Prudhoe Bay, it's, there are no roads. And so the pipeline company would have to build a road. And sort of being an adventurous type, I talked to my advisor and said, we should do a biological survey, and this is February, of the road, and by following the bulldozers. And he looked at me and said, well, that's an interesting thought. Let me talk to it about the Institute director, who thought it was a fabulous idea. And, almost overnight, a few days, they committed 3 Dodge Power Wagons, big pickups, and 6 of us. And we loaded up with fuel and food and whatever it needed to survive, and went off on this expedition. Now, again, it's February. It's a little chilly, minus 40, minus 50 degrees, and we were camping. And so off we went, driving, brought our own fuel, etc. Some people slept in the trucks, others of us toughed it out in the tents. That may explain my problems today, I don't know. But we followed about halfway up, across the Yukon River, which was frozen. Scared me to death driving across the river, but it was 3 feet of ice, so it was really nothing to be concerned with. But the banks on either side were like 10 feet tall, and you had to get a running start and sort of bounce up them. And I don't know what we accomplished biologically, but we certainly ended up with some memories. And we discovered a hot springs, which was unknown, which I enjoyed the opportunity to take a leisurely bath in. It was interesting. A lot of flies along the surface of the water, and if they were disturbed, they'd fly up, and they'd chill, and they'd come crashing down because they couldn't handle the low temp. Because just a matter of a few inches above the water is minus 20, 30, 40 degrees. But that was that activity. Also went off on a cruise in the Bering Sea the next February. Came home and bought my wife a handgun, and I told her I was leaving for a few months to go on a research vessel out of Scripps Institution of Oceanography and an icebreaker, and to study primarily mammals in the winter on the Arctic ice. And while most people won't appreciate what I have to say next, I was the marksman, and it was my responsibility primarily to acquire the seals, the walrus, etc., and so that the nerves could be extracted and so forth by my advisor. And we did that for a few months, and that too was a rather interesting adventure. So I threw in a little extra trip on there. I mentioned earlier we also had a 5-year program in the Antarctic, and we went there typically December, January, February, sometimes March, and that was their summer. And we went to the Arctic Peninsula. There's a research station, Palmer Station, run by the US Navy and National Science Foundation. And we were there, again, studying the only insect species of the Antarctic, a wingless fly. And you might ask, if you're a bit naive, why is it wingless? And the answer is, where would it go? A little chilly all over the place. So, next question.
Well, yeah, you certainly explored. As my mother has often said, it's amazing that 1, I or my sisters were born, and 2, that we had a father growing up our entire life with Big John's explorations, because those Antarctic journeys happened while I was in middle.
You caused a problem in my life. I was not eager to get involved with procreating, and in our first faculty position, we had a farm, and while in Alaska, I got a pilot's license, and so I bought an airplane, tail dragger, Cessna 170 clunker, and I used to land and take off in the backyard, and I enjoyed it immensely. And whenever we flew anywhere with my wife, she'd be in total panic. So I kept giving her the maps and asking her to look out for certain landmarks like radio towers and high tension wires, et cetera, which weren't there, and she couldn't find them, but she was busy. When John was born, she came home from the hospital. She says, "Your flying days are over. You've got to stop flying. It's too dangerous. You have responsibilities." So I flew down to the local airport and put a for sale sign on the plane, and a week later sold it. And I've been bitter about that experience ever since.
That's interesting. He keeps asking me when I'm going to buy him a new jet, and not a Cessna anymore. Now it's got to be a jet. So I got my work cut out for me.
Oh, there's some Cessnas I might like.
Well, okay, right, okay, gotcha. Oh, going on that was great. I'm going to shift gears, I would say, to let's go to the last 3 decades of your career, and shift into the clinical side, the follow on of where you launched. You started talking about, you got involved in cryosurgery and cryopreservation and the corporate world, because I think there's a lot of growth and interest in that space now within, well, all members of this field. I was going to say young, but really everyone is kind of getting into that area or is in that area. And, your team was the first to describe the molecular response of cells to freezing injury, and really start to dive down into it. I mean, two-part question there. One, when your team got into that work, what did you feel was the significance of the discovery at the time? Or did you feel it was a significant discovery at the time? And two, how did you get into that area? How did you approach moving on to that, molecular response to freezing injury path?
Well, I started out really with a base of knowledge that was consistent with the entire field and was very naive. The idea that if you apply a low temperature in the field of cryosurgery to a tumor, you will kill it. Well, that's true in some respects, but you don't kill everything. And there's a thermal gradient between the probe, which is placed in the tumor, and the periphery. And the periphery is at the nominal melting point, slightly below 0 degrees C. And so there was always a chance for recurrence of the disease. And so what was done to overcome that was to freeze beyond the margin, and typically a 1 centimeter positive margin. Well, 1 centimeter doesn't sound like much. So if you're treating a 3 centimeter lesion, and so, okay, you go out to 4 centimeters, if you calculate the volumes, most of the tissue targeted ends up being in that peripheral zone. So there's a lot of collateral damage that can be done. So the question became how do we sensitize the tumor to the freezing process? And at that time, this is the early 1990s, I mean, the idea of cancer, it was sort of a unique system. It was one kind of cell, kind of dominating, uncontrolled growth, et cetera. None of that was really quite accurate. And it's a very organized, the tumor is a very organized tissue, with multiple types of cells, all with different freezing sensitivities and all with different responses. And of course, there's always cancer stem cells, which are pretty much immune to the freezing process. And so we were faced with this conundrum of how to address this problem. And so the first idea was is there a way to sensitize the tissue to freezing? And we began by trying a number of chemotherapeutic agents at very low doses, just stressful. And they worked really wonderfully in sensitizing the tissue to the freezing process. So one, instead of having to take a tumor to -40 or -50, which can be difficult, you need liquid nitrogen devices and not argon devices. And, different hospitals had different devices. So the outlook for success was quite different if you understood the process. And so with sensitizing agents, you can move all the cell types to a freeze sensitivity between -10, -15. One of the important sensitizing agents we looked at, and some equivocal nature of the data, is vitamin D3. And that seemed to have a fairly significant effect. And of course, that is not inherently a cytotoxic agent. So that offers promise. The difficulty in those discoveries is that cryosurgery is not a first-line therapy. It cycles an interest in and out, and it's simply thought of as, ice cube. And there's a whole cell molecular biological series of events that are going on. I mean, we first reported that in the elevated subfreezing temperatures, apoptosis occurred. It was unknown. It was the early days of the discovery of apoptosis in mammalian systems. And then went on to describe different categories of apoptosis, necrosis, different categories of necrosis, and so forth. So there became a whole hierarchy of cell and molecular biological activities involved, and really had to make a pretty major shift in careers from insects to cryogenic physics to cell molecular biology. And luckily, I had a really diverse science background, and I could, walk into those fairly comfortably. Does that answer your question?
Yes, it does. I think that, if you could expand on that a little bit and flip the coin over to the cryopreservation aspect and how the molecular work your team has done there, it's kind of, if you will, in my words, altered the landscape of cryopreservation. I think that would be interesting to the audience as well.
Yeah. Yes. Well, and again, cryopreservation has sort of been viewed through the tunnel vision, and I don't mean to be negative on it. I'm recalling a quote by Harry Merriman in the preface to Advances in Biopreservation, where he said the problem with the field of cryobiology is it made its initial discoveries too easily. That is, red cells, and of course, when I was in college, they were not cells, they were corpuscles, because they're really megakaryocyte cell exports, and sperm, which again is a cell export. And those cryopreserved readily with glycerol and DMSO and so forth. And beginning with the work of in Polger's lab in the UK in 1949 and on, it seemed like quite simple. You adjust the biophysical environment of freezing to limit intracellular ice and to limit too much extracellular ice, and the cells will survive quite readily. Roadblock was hit when they moved to nucleated cells, particularly a variety of types of lymphocytes, some of which I don't think are really that cryopreservable today. And the field went on, and it stayed with that mantra of cryoprotective agents. And DMSO came about in the late 60s by Lovelock, and that was the silver bullet at the time. and had been for many years. But again, as we begin to look at when you cryopreserve cells and you bring them back, this is a substantial population that even in the best of preservation procedures die off. And most users of these cells will allow a few days for them in culture, quote, to recover, unquote. Well, if you look at those few days, a lot of cells continue to die a day or two after thawing, and some begin to procreate. And so you come to a stable cell population, usually 3 days or so afterwards. And so really the question was, why does death continue, et cetera? And what we found out is that cryopreservation or the freezing of cells and the chilling is as much of a chill insult to the cell as anything. Because if the cell actually, mammalian cell freezes, it's dead primarily. But it dies from extensive chilling. And when we looked, we saw that, again, apoptosis was involved. And I think we maybe were the first to report on that in cryopreservation. Again, different levels of apoptosis were launched at different sub-freezing temperatures, and then ultimately necrosis if one went low enough and if the cell was not to survive the freezing process. So again, cryopreservation is much more involved if you wish to understand the molecular basis of it and ultimately improve it over time than just the physical events associated with ice itself, in my view. Next question.
No, that's a very good view. I think that's shared today. It's interesting you brought up the 3-day recovery. There was a webinar that I attended yesterday, and Dr. Hubble, Allison Hubble, current president of the Cryo Society, she actually made the point that in a lot of stem cell therapy applications, there's a question on repeat freeze, thaw, and recovery, and she said that there's no standards yet, but if you will, the guidance is to allow 3 days recovery so that the post thawing before use or refreeze, or this was in the laboratory, not necessary for injection. And it was because of exactly the points that you raised of that cell population is stunned, damaged, injured after freezing, and they may not necessarily do what you want them to do in the lab for expansion or the like at cell therapy, especially when you get into T cells, NK cells, a lot of these more sensitive cell systems, as you mentioned. So it's still an issue today that the field and researchers are facing despite the advancements. And so I think that the molecular work that you and your team started, back in '98, I believe it was, is still, whether they want to admit it or not, guiding the field today. So that's really important. One of the questions I think on the global side of things is, how did you approach translating basic science research to the clinic, using your knowledge or your research and getting it to therapeutic tools? You mentioned both the preservation and the surgery side of things, and products came out of it. That is not common for academics, frankly. They get all the research money and don't produce much, but papers, and that's the trade. How did you make products? I mean, you've made at least 2 cryopreservation products, one of which is now the gold standard in cell therapy industry, and well, frankly, reinvented the cryosurgery field. And so, those devices, those probes, how did you do it?
I don't have a clue.
Oh, okay.
Oh, no. I just followed my interests. And when I found myself in a particular situation, I just invested whatever knowledge I had in the project, like you're mentioning, cryosurgery and tools. I mean, when I joined that company, which was to be on a temporary basis, I ended up moving on to be president of the company and selling it. But the devices that existed at the time were substandard, and we just went, in that case, into the science of cryogenics to look and understand, if you will, two-phase flow and cryogens, et cetera. I mean, stuff that typically only physicists get involved with. I did have PChem in college, which sort of helped me. And we were able to, through a lot of trial and error and ignorance, move and progress forward. And one hand in my lab, we had, really, I would say, physicists and biophysicists, and the other hand, cell biologists. So I was really happily positioned in the middle to benefit from the work of both. And I could simply tie it together. And tying that together allowed for, support. We had long-term NIH support and so forth. So I simply followed the lead in my mind.
Okay. Along that line, I mean, you're often referred to as the father of modern cryosurgery. What does that
By who?
By who? I mean,
Come
anybody
on.
in the field of cryosurgery, basically. What does that mean to you?
It means that someone is applying a label that probably is best applied 20 or 30 years from now, if still relevant. I mean, in science, there's a lot of fathers of, not too many mothers of, but that'll change. But, you sort of lead in your own way, and sometimes people follow. I know I made significant contributions, or my lab has made significant contributions, and I'm sure some of them are on there. I can't read the screen very well because I'm sitting outside. Whoops. Hello? Can you hear me?
Yes.
Can you see me?
Yes.
I can see nothing but the word Zoom.
That's all right. Just keep going. You're fine.
Okay. So that just doesn't sit well with me. And so if I'm the father of cryosurgery, does that make you the son of cryosurgery? I don't think so. And it's not a label, I'm belaboring this, that fits well, personally.
You see, there's still lessons you teach me in humility. I will write that down. So you've been very successful bridging academia to entrepreneurship. And so I think that one thing that, I get asked a lot, and I don't have a good answer, and maybe you do, is, what advice would you give to researchers looking to bring their innovations to market, maybe to come out of a postdoc or a faculty position, let's say, and move into industry? Is there any tidbits of advice, any John Baust rules of success in entrepreneurship within the field of, well, I'd say cryobiology, but any field could be applied, wisdom that may be able to help the next generation?
I wish I had some sage advice here. It is difficult. And I think, as you know, trying to raise funds for your company, it's very difficult. And if you have a technology that's unique and highly marketable, you probably have one chance in 100 of raising sufficient funds to pursue that. You may find a company willing to partner with your technology, with you being a minority partner. The scientist is always going to be a minority partner, and the scientist most always will get the raw end of the deal financially. That's the way capitalism seems to work. But my advice would be make sure you have a good patent, which is pricey and difficult to do, and try to find, if you're at a university, through your IP department, hopefully the right group by which you can make the right kind of contacts. I know I tried to raise funds for Cryomedical Science, that first company, and it was the 100th investor group that I met with that I succeeded. Now, that may not sound that significant, but you know how many trips to Manhattan and San Francisco and Switzerland, et cetera, one has to make, and how many presentations and have to undergo trial by fire. An awful lot. It's an exhausting process, and most people would give up, or they'll find a deal that's probably not to their liking. So all I can say is if you're an entrepreneur and you have something that you believe is of value, walk carefully, rely on experts beyond your skill, particularly in the business world, and hope for the best.
Very good. And for everybody on the Zoom call that is under the age of 55, when Big John said 100 meetings, those were in person. There was no Zoom. In fact, there was no internet for the masses then. So those were all plane trips, phone calls, this thing called a fax machine. So today's equivalent is about 1,000, is what they say. It's because of Zoom and virtual meetings has increased tenfold. So it is a long slog. Let's see. I guess there are a lot of emerging areas in science, and we're not kidding anybody. Cryobiology, cryo is not a glamorous field. What do you see and I guess it'd be in two different areas, the surgery and the preservation, but what do you see as the current and future role of cryobiology in clinical medicine?
Well, I wish I had video here. I don't have a picture. I would show you what I see. And I'm not sure this will work. I will lift up my laptop and show you my environment.
We
Does that
see
work?
it. Yes, it did.
Yes, I'm retired in the Florida Keys, quite happily. No, but to seriously, what is the future? I mean, I think there's a lot of future. One has to, I want to go back for a second, where we're talking about that delayed cell death, because it leads into answering this question. I seem to recall a figure, and it may have been from Alison Hubble's work, where they were trying to develop a therapy with lymphocytes, and they were injecting them post-thaw, and the therapy completely failed. And it wasn't until 3 or 4 days later that the therapy would work, and that's because there was a new population of cells developing. So, so the future is, first understand what in fact the parameters are with which you're working. And the immediate areas that I think need attention is clearly organ preservation. We're trying, and there's a lot of good leadership involved in that, but it's not a simple process. Just because a frog, I'm not trying to throw stones here at Ken's story, although I'm sure he'd throw stones at me if he had the opportunity. A frozen frog heart has no relevance. Little relevance, I guess, is a better phrase to use as a scientist. And versus an animal heart. I remember a very long time ago, I decided to try to get into organ preservation. And I started with hearts. And I had an endless supply of dog hearts because the lab above me worked with dog kidneys. And when they were done with the dog kidneys, we could have the dog hearts. Well, I had zero success. So there was an expert on cardiac function and so forth. And I went to see him. And he says, well, you're never going to have success with dog hearts. They're too sensitive. You need to work with sheep hearts. So the message there is really, to begin with, working with the wrong species. And the second message is species count. And so if I were to have had success with the dog, I'm not sure what that relevancy would have been. Certainly would have favored granting agencies. So organs are really important. And and I recognize that more important as I age. The cell therapy market, I think, is still very open to development. And to get involved in that, one really needs to help an early stage company or startup company solve some of their problems. Because once they establish their methodology and they have their FDA registration, they're really not going to change. Even if you could show them better outcome, they're not going to change. I remember years ago, we went to a certain cell preservation organization in the Virginia area and showed them enhanced preservation of about a dozen of their most marketable cell types. And the CEO got really excited. He says, we really need to get involved and understand this. But the CFO tapped him on the shoulder and said, excuse me, what you're telling me is that your technology will improve cell preservation, the cells that we market, so that we have two qualities of cells. One which represents 25 years of collection, which is inferior, and yours, which is superior. That ended the meeting. And that, while that's a long-established company, you'll find that in any group along the way. So again, cell therapy market, lots of opportunities. John mentioned earlier Svalbard, Spitsbergen. These are Arctic islands north of Norway, which I spent a couple of seasons working in. And of course, they've developed not in the glacier, but in the mountainside, which has a steady temperature of something like minus 20 degrees C, a bank. And they're banking everything they can. And those of us who know a little bit about cryobiology know, except for some seeds, plant seeds, that's a dead end. A lot of what is being banked will not survive. Now, certainly the DNA may be preserved and used later on in the event of, a world-ending event. But again, I think there are massive investments being made without the basic understanding of the science that's behind them, or the lack of science, or the poor science. So I don't know. Does that? It's whatever cell type, whatever mammalian tissue, primarily human, that can treat a disease is marketable and represents an important future of cryobiology.
Okay. couple more questions here, just as we get close to time, about to wrap up. But could you expand on that same topic? So basically, where do you see future opportunities are like on the cryosurgical side of things, cryoablation?
Oh, I don't know. That's a toughie. The field began modern or neo-modern in the 60s, and it showed a lot of the success, but also showed major complications. It was actually quite effective. It really began with prostate cryosurgery and liver cryosurgery. But the collateral damage that was done to patients by overfreezing was enormous. So it disappeared. We tried to bring it back, again, through the company Cryomedical Sciences, because we had the investment to do that, and invested just under $100 million in trying to get it reestablished in the medical field. And we placed devices and trained and so forth. Just about 200 hospitals, medical schools, US and Europe. And people were very happy doing it. The problem became it wasn't a technology that was taught in medical schools. And by the time we finished, it started to be in the medical textbooks, but sort of as, an ending paragraph in chapters on surgery. So if physicians are not trained in the technique early on, it's unlikely that they'll move into a field, because medicine is extraordinarily conservative. I mean, a discovery today can take 10, 20 years to be realized in the clinic. And in fact, with the advancements today, it probably won't even be realized. Something will surpass it for another 10 or 20-year period. So the future of cryosurgery, I think, is limited. I think what's needed is the development of cryosensitizers in particular cancer situations, like in the prostate, and many have been developed from a basic sciences perspective. They need to move to the clinical level. And the challenge there will be get funding. And surgical technology is not in vogue today because of the return on investment. It's not that significant. Okay.
I mean, yeah, it's not great news for me, but no, I think that I understand what you're saying. I think that with Boston Scientific and Siemens coming into the landscape on the cryosurgical side with the acquisitions of Galil and Endocare respectively, we are seeing a change. But on the investment side, you're absolutely right. There is, for companies, there's a challenge there. So that certainly.
Well, it's important to recognize that while those companies acquired the other competitors in the field, what are they going to do with them? What they've acquired was a list of surgical sites and some equipment technology. Are they going to market that? Or are they going to look at maybe substituting that for other technologies and not putting the investment into reinventing? One of the companies you mentioned that they purchased, it developed its platform in the early 1990s, and it never changed. And the second company, Galil out of Israel, which has been through a number of ownership changes, again, was in the later 90s, and the technology hasn't changed. So the state of the art today, from the commercial perspective, has been the acquisition of what I would call past generation devices and technologies. are these companies now willing to make the expenditures to invest in what you have or the next generation of technology? The big unknown. I hope it works.
That makes total sense. Got two more questions. First, I got a question from the audience in the chat. So Kristen asked, she's a tumor biologist working on cryoablation of subcutaneous tumors in the veterinary medicine space, and they're always looking for better ways to preserve the skin distal to probe insertion, so less damage there, while optimizing the freezing of the tumor mass. So with your discussion of, insects producing glycerol, trehalose to better survive cold temperatures and other cryoprotectants, her question is, do you think that applying CPAs, specifically glycerol, trehalose, or other, would be good topical protectants for protecting the skin during a freezing procedure?
It should. Right now, I mean, at least in my experience in dermatology, is they'll simply put a saline soaked cotton swab, piece of gauze or something on the surface of the skin to help buffer the temperature decline. So certainly, glycerol will do the same. I don't think dimethyl sulfoxide is what one would use, because that's so permeabilizing that it too would get into the periphery of the tumor during the process and probably might act to do a little preservation. But of course, dermatological applications are readily done with cryoablation. I have a plethora of experience in the veterinary field. Not really, but let me tell you one side story. We got contacted by the University of Pennsylvania vet school, and that there was a horse named Campfella, quarter horse, which was the highest earning horse of all horses. I didn't know the quarter horse field at all, more so than thoroughbred racing and all that. And it had a tumor in its testicle, but only one testicle descended as a colt. And they were making millions a year on breeding this horse. And the vets had advised the owners simply to put the horse down or to cut off the testicle and put it out to pasture. And they thought, would cryo help? So I went to the horse veterinary school in Pennsylvania, and I brought with me a noted urologist who froze prostates. And here is this horse hanging by its hoofs, moved into a horse operating room, which is an entirely different concept. And team of surgeons, and we were part of it, and he was to freeze this seminoma. Well, the man totally panicked and really couldn't do anything. He was just at a loss. So nothing happened. Came back the next week with a different urologist who could handle it quite readily. And we froze a portion of the testicle, had the cancer. Well, 6 months later, they called to say, well, the cancer was back, but they were still able to collect semen and were still making lots of money. And so actually we went back, and a third time I did the freezing, probably go to veterinary jail for doing it. But we extended the breeding of that horse, must be 5 years, to the betterment of the owners of many millions of dollars. And finally, the horse then was castrated and put out to pasture and lived happily ever after. The interesting part of after the surgery, the horse recovers in a swimming pool. They have a special four-legged float that they put the horse into because they will jump around and thrash and basically injure themselves. So there's a pool, and B.F. Goodrich made this special the float for them, and they would swim for a while until they basically recovered, and then they'd be lifted out and put into a padded room to complete the recovery and so forth. Another interesting experience in life. That's all. So dermatology, subcutaneous tumors. I think if you're concerned with skin death, so forth, after the freezing process, topical application of something like glycerol should offer some benefit.
Yeah, and I can repeat, Kristen, we have been doing some work in the breast space, and we worry about breast cancer space, and we worry about skin damage and freezing from a cryoprobe. And what we found is either a saline-soaked gauze, or a 20% glycerol mixture soaked gauze with topical application just right around the area you're worried about freezing. Works extremely well. So at least in our hands in the laboratory, we haven't done a lot of veterinary applications, but nonetheless, I can speak firsthand to what Big John said. We've seen that work very well too. If anyone else has any questions, please feel free to put them in the chat. I'm going to wrap up here with one final question. Well, it's a two-part question, but first of all, I guess it's one and the same now that I think about it, but if you could say what part of your work in cryobiology you're most proud of, and looking back, what do you hope your legacy will be in the field? I guess it's a three-part question because the last is, what's left on your to-do list? So what are you proud of? What do you hope your legacy will be or is, and what's left? What's next?
Well, here I am in the Joe Biden stage of life, and you're asking that question. I've had good students, graduate students, who've done the work, who deserve much of the credit. And depending upon how they go on in their future, they will get it or not. That's the advantage of being a senior professor. You tend to harvest the benefits of others. I think all of it to me was important, and I really can't pick any particular area. I was certainly happy to recognize that there are hundreds of thousands of people who've been treated with the technology I helped develop in my students who are cancer-free. I mean, that's a great little thing to put on the back of a tombstone, I guess. I'm also, very content with what we did in the polar regions. But that, of course, is really more personal. No one reads the papers that are published in the 70s. My first paper was in the 60s as an undergraduate. I'm not even sure the journal exists. Libraries in the basement are filled with volumes of archaic work. I'm sure the investigators, as I am, are proud of it. That's what we did. Science moves at a pretty aggressive pace these days. And what was the balance of your question?
That's a good question. Now I forgot. So it was like, what are you most proud of? What do you hope your legacy will be? And then what's next? What's on your to-do list?
Well, if I lift my computer, you can see around, correct?
Yes.
So if I do this, that's my next agenda item, the pool.
Okay.
No, I really, in science, I really don't have a future vision. You and I will obviously talk from time to time, but I've become fossilized, and so that will be limited. We're also separated by a very wonderful climate and a brutal climate. My legacy, I guess the legacy will be short term. It'll be citations, people who look at the work we've done, read it, maybe cite it, and so forth. Today, I get a report from ResearchGate every day about how many people have looked at a paper, what the paper was, how many have been cited, et cetera. It's part of the generation of garbage information. I mean, I don't really care or need to know who in Tajikistan cited a paper from 1996 in cryosurgery, but that's the kind of information I get. So I think any of our legacies will be that, and I've never thought of this question, but it'll be the paper record.
Well, I think it'll be more than that. I think, as you said, the thousands or hundreds of thousands of patients' lives that you impacted and their families' lives.
But they won't know me. They have no idea of my name. They will thank the surgeons that basically saved them. The scientist doesn't get the credit there.
That actually stimulates one more question in my mind, because I often say this, and a lot of my colleagues, our colleagues, frown when I say this, but you're absolutely right. Cryobiology is not glamorous. It's a tool. It's a tool to enable other fields. Would you agree with that as a holistic statement from a surgeon's perspective or a cell therapy perspective?
Well, yeah, in part, it's part of it's a tool, but the other part, I think, is the in-depth on it. We still don't have a really in-depth understanding of mammalian systems once you begin to chill them and expose the freezing environment. There's a lot more to it than we know. And, until it can be as routine as applying a bandage, there's work to be done.
Couldn't agree more. All right. Any other questions from the audience for today? This has been excellent. I certainly learned, as I said, every time we talk, I learn something new, and both on the scientific and the personal side. So it's always cool. And it was, I want to thank the society for giving me the opportunity to do this, because there aren't too many father-son, parent-sibling or parent-child teams out there in the world of science, and hopefully I'm doing a better job than Shredder Sanders is and carrying on your legacy these days. But yeah, okay, we do have a question come in. Okay, here's this is going to be a controversial question and topic, and feel free to answer it as free as you would like. The question relates to whole body freezing, cryopreservation, and with different organs having different outcomes in cryopreservation. What are your thoughts on whole body cryo, and is it possible? Knowing that anything could be possible with science, never say never.
Yeah, I can't comment on what might be possible in the future. I mean, I've seen that Spock was carbonized, right? Star Wars, and they can bring him back from that state. not really. Um, I've never been a proponent of whole body freezing, in part, and I've argued a lot with the cryonicists, and I've even visited their laboratories. And I remember walking into one lab which had a liquid nitrogen door, which was 10 feet across and about 10 feet high. And in there, they were proud to tell me they had, 3 patients, 7 heads, 2 dogs, and a cat. And I just said to myself, so that's the state of this field. That was a long time ago. We don't have the technology to accomplish that today. And I'm not sure what we would be attempting to accomplish. The idea is to freeze the body or freeze the head, which is part of the marketing, and ultimately be able to reanimate critical cell masses or something, and then reconstruct the body around that. Is that possible? Do we have saber-toothed tigers and mastodons roaming the streets of New York and London and so forth? I don't know that that's a particularly good idea. And I don't know that that's going to happen. So I'm extraordinarily skeptical of it. I have not signed up personally. I don't have any intention of signing up for that. And if we left the development of this technique to Kelvin or Mike or someone, if they use the same progress they are with hearts and other organs, and they have to do the whole body and all the different organs, it's going to take forever. And I mean that respectfully. It's not on my horizon of recognizing the ability to succeed in doing that. So therefore, I do not recommend it. I understand the fee is like $100,000 or $125,000. I would recommend taking that and going on a hell of a trip.
Well, I think that's some pretty sound advice there. Yeah, the other side of that in the cryonics world is, they are, not we, I'm not doing it either. You're freezing sick people. So not only do you have to freeze somebody and bring them back, you got to freeze somebody that was dead and bring them back, and you got to freeze somebody that had a terminal disease or died and come back. So there are more challenges than just the freezing once you get them thawed out. But
those are trivial.
yeah, we'll have those figured out in the future.
Sure.
So, all right. I don't see any other questions coming in. I mean, do you have any final thoughts? Anything that we missed or didn't cover today that you'd like to share?
Gee, covered a lot of territory, pole to pole. I remain a strong proponent for the field of cryobiology. I think it has a future. I think in some areas, the focus needs to begin to shift slightly. There's a lot of hard work ahead, and but I think success will manifest itself. So good luck in the future, fellow cryobiologists, while I lay in the sun.
All right. Well, thank you so much for your time today. I really appreciate it. You can get back to going in the pool. I thank everybody.
Where do I send the bill?
You send it to Nicole.
Okay.
Yeah, just hit reply to the email with the link in it. She had to sign off, so she'll get a real good kick out of that. Or email me in panic. But I thank everybody for attending today. I really appreciate it. Thank you for the questions. And Yeah, we hope to see you at Cryo this year. Unfortunately, neither Big John or I will be there in person, but we'll be Zooming, so virtual. You can always, oh, I got a message. So, oh, thanks, Mike. Appreciate it. Mike says good to see us both, and he enjoyed the talk. But yeah, so, and if you ever, have any questions, want to reach out, feel free to reach out to me, and I can forward your, I'm going to put my email in the chat. I'm not strong enough to type and talk at the same time. We can try and answer your questions, get you in touch with Big John and the like. So with that, I think I will bring this to a close, unless there's any last minute questions, which I don't see coming in. So everyone, thank you again for your time. Enjoy your day, afternoon, morning, evening, because we've got people from all over the world still on. So have a good one, everyone, and we'll see you next month. We have not announced who's speaking next month yet, so keep your eye on email blasts, LinkedIn, X, possibly Facebook. I'm not on Facebook, so I can't answer that. For the announcement to go out and on who's going to be doing the what'll end up being June, because we're at May 1st now, so sometime in June. Enjoy the rest of your day, and we'll look forward to seeing you at Cryo 2025. Bye, everybody.
Bye.
Thank you very much, Big John. Really appreciate it. Great job.
Okay. So I just close this out.
Yeah, you can. I'm going to hit end. It's going to end it for everybody. So we're just looking to see. There are a couple people still around, seeing if any other questions popped in. But I think we're good. So thank you so much. Have a great afternoon. Go change and take a dip.
Okay.
Bye-bye.
Bye.